Hello, welcome back to the last segment of the videos talking about the analysis of the tallgrass prairie in Iowa. What we've done so far is talk about the point pattern analysis, and now we're going to try to link that to what's going on in the plant community, which was one of the main drivers of the study. I'd like to acknowledge that a lot of the pattern analysis was actually done by a master's degree student who worked for me, Ben Kloss, and what I'll talk about today has a lot to do with work by a PhD student, Kelly Wolf-Bellin, who did a detailed plant community and soils analysis, which we tried to link to the Gopher mound production information. So here's a picture of Kelly working in the field collecting plant community data. This is done in a very sort of standard way for this kind of analysis in systems that have this kind of stature. We have a frame made out of plastic here. There's a bar in the middle that you can see that has small holes drilled in it, and she drops what we call pen flags, which are basically wire down through there, and determines what plant is touched by the first drop of the pen. She does this 100 times over each sample site, and this gives us cover data with a fine grain division of 1% cover values, which is good quantitative data for this kind of analysis. So this shows how we sampled within the grid. So this is the same grid system that we had for the point pattern analysis. We have two different things that we're looking at. We're looking at community surveys and we're looking at soil surveys. These were placed in a set position within each of the 10 meter by 10 meter areas. Within the green squares there, the community analysis was done with the pen flag drops that we talked about. In the soils, we took soil samples and had them analyzed back at the university. This just gives an idea of the things that we measured. For soil variables, we measured soil moisture, pH, NPK, total carbon, and carbon to nitrogen ratios. For the plant species, we had 86 species that were observed in the entire study, indicating to some extent the diversity here. 71 of those were forbs, that is non-grassy herbaceous plants, and 13 of those were nitrogen fixers, which are really an important component of these ecological systems. In the analysis, we didn't take any information from all of the species because some we knew were there but were never hit by a pen flag. They didn't enter in. So in the overall analysis grid, there were 44 species, but in the part that we studied, which was only where there was gopher activity, there were 38 plant species involved. So with these data, we did a typical ordination analysis. Hopefully some of you are familiar with these. Basically, it takes the soil data and compares it to the plant species and comes up with general gradients that explain the distribution of the plants. And so what we can see in here are measures of individual species. So that's Astragalus canadensis, which is a species in the system, and the placement of it where that X is basically the point where it's found on average in terms of the ordination. What the ordination is two axes that are perpendicular to one another. This first axis, CCA1, captures the most amount of information that's contained in the soil data that is correlated with each other, so nitrogen, phosphorus, everything, how they change in concert. And so this represents the primary environmental gradient that's potentially influencing the plants. CCA2 takes what's left of that information and looks at the maximum explanatory power in the next dimension. And so what this does is suggest that if there's a trend in the distribution of plant species, it can be coupled to these two gradients that are related to one another, sort of on the X and the Y axes. What we see is that species that are circled in green over on the left-hand side are species that are found only in low-lying areas where it's quite moist. And what we can also do is take information about elevation. Remember, we did the survey. We also used that to get elevations from each gopher mound. So we have a really good elevational map of the site. And so what we find is that there are two general gradients that we can pick out relative to elevation and disturbance. Elevation goes in that direction in terms of explanatory power. So the way to interpret this is low values are to the left, high values are to the right. And so the plants on the lower left side would be at lower elevations, and the ones at the upper right-hand side are at higher elevations. This is correlated very strongly with the two underlying nutrient gradients, so it's the primary thing that's structuring the system here. What's really interesting is that if we take a look at disturbance, it runs in this direction and is perpendicular to elevation. So it means that plants that are found in the lower right are experiencing much more disturbance than plants in the upper left. And so this represents structure that's induced by having different amounts of disturbance on average in a different location. We sort of corroborate this interpretation because if you look here, this is bare ground, which was also entered into the data, and it is at the extreme lower right, which is where disturbance rates are the highest. So this really makes sense in terms of sort of suggesting that our interpretation is correct here. And so we can take this information that's obtained from our gopher mound analysis and the way we interpret it, couple it together with the analysis of where plants are distributed and these primary gradients basically related to soils, which are correlated with elevation, which is the main driving gradient here, and we can put the two together. And so from this, we can draw some overall conclusions that can be explored more fully in subsequent studies. And so the first point that we can make is that in this last analysis, soil variables are strongly correlated with elevation, and this forms a primary environmental gradient. Gopher mound production exerts a significant influence on the structure of the system that's independent of the primary environmental gradient. Again, this is because disturbance falls out as perpendicular to the elevational gradient, which is the main descriptor of what's going on here. And lastly, the influence of gopher mounds is independent of the, well, I've already stated this actually, is independent of the primary environmental gradient. And so at this point, this, I think, shows how we can use a point pattern analysis and couple that to another ecological analysis to get better insight into how organisms are structured in these systems, particularly when there's something like disturbance that's playing a large role. I think we've learned a lot about how to apply some of the techniques such as Ripley's L to get an understanding of the disturbance regime and different components of the spatial scales that are involved at different time periods and within these territories. I think we could gain a much deeper understanding if we applied some of the newer techniques that exist now, such as pair correlation analysis, which gets rid of some of the problems of L analysis, which you've probably heard about. So I hope you enjoyed the videos and that they've given you some insights into how point pattern analysis might be employed to study some really interesting ecological questions. And it's given you sort of an idea of the development of the field as well, thinking back to the videos where we talked about different methods that have been used to measure locations and place XY coordinates on particular points or processes that have occurred. Thanks a lot, and I hope you've enjoyed the videos.